So with all this experience, I think it's very important to have a sense of community for the human development in any area that you work, in anything that you do. And particularly in this field that we are at, the bioinformatics field, it is also important to teach kids and adults computational concepts because it's a form of investment in their lives. As we said, I come from an undergraduate background where I didn't have any exposure to computational programming. So it's really important to provide people with that type of training in a field of science that in my mind, and I guess many of you would agree with me, is so needed these days. So for many of us, and for me, particularly, a computational algorithm is a thing that I never used it to think. 5 years ago, for instance, before that, I never used it to really think about what a computational algorithm is. But really today, I would like to give you a very general idea of what a computational algorithm is. It is basically a set of rules to be followed in calculations to obtain results from as an output from a given input. So you basically have some piece of data, you give it to a computational algorithm, and you get a result out of it. And thank you, Elia, so much, because you talked about this concept of classification, how it is so important in cancer and in these advanced genomic studies. But really, I like to start from the very simple principles of science. And I guess we have been using these some sort of classification for a really, really long time here. This example of classifying, for instance, animals. So we can, for instance, think about classifying the animals based on their number of feet, and I hope I'm saying that correctly in English. So here we have animals with 2 feet, and here we have those animals with 4 feet, and this is a system of clustering or classification. We could argue that in that regard, classification system, like classifying the animals by their species, is a more refined way of classification compared to clustering. But really, we are talking about grouping subgroups based on similarities that they have. So how can we get started with this computational classification, I would say, that we are so interested in? First, I would say it is really, really important to identify a problem. As I am going to tell you, the problem that I work with is cancer, particularly neuroblastoma, which is a pediatric cancer. The other thing that I would say is important is to identify a sponsor, so a mentor, a school or a group, a science group that you might be interested in. And really, what you need is someone or an institution that has the potential to guide you through the process. And frankly, I joined OmicsLogic a couple of weeks ago. I'm still getting used to all the resources that they have, but so far, I think the resources that I have seen and have tested and used, they're very, very valuable resources in the bioinformatics field. So I said that it is important to identify a problem that you're interested in, and the one that I told you I am interested in is neuroblastoma. It's a complex or polygenic disease. I'll not go too much into details, but I want to say that it's a problem in the development of the sympathetic nervous system. So the sympathetic nervous system is this part of the nervous system that connects organs to the central nervous system here in the spine, for instance, as you can see. The other problem that I want to go to in neuroblastoma is this concept that we have 2 major populations in the tumor. So we have the whole mass of the tumor. We could argue that there are many subpopulations, but I'm going to tell you about the 2 main subgroups, the 2 major populations, which are the population of adrenergic cells that look actually like neurons. That's why you see these projections from the cytoplasm of the cells, making them really look like neurons. And we have the mesenchymal cells, which do not have these projections, they are not, they do not adhere, sorry, they adhere very tightly to substrate. That's why they have this name of S-type, which means substrate adherent. But the difference between these cells is that the adrenergic cells are really differentiated and the mesenchymal cells are not differentiated. You can also think of them as cells with a stemness potential very high. So these mesenchymal cells are not differentiated. They are not sensitive to chemotherapy. They do not die upon chemotherapy. And they lead to this concept of minimal residual disease, which is the concept that after you give chemotherapy treatment to the cells, the chemotherapy is going to kill the adrenergic cells, but not the mesenchymal. And the mesenchymal cells have the ability to differentiate into adrenergic cells. And that there we have a problem because the chemotherapy is not going to kill the cells. And so there is a chance that the disease is going to come back. And when the disease progresses or relapses, there is an enrichment of the mesenchymal cells. It is really important to classify neuroblastoma tumors, and I'm going to give you a more detailed idea of what that looks like. It's really important to classify the tumors into adrenergic or mesenchymal because that has a potential for prognosis. But so far, I mean, that's the part of research that I do. Here, this slide shows you what really the ultimate level of prognosis is in neuroblastoma. So a neuroblastoma tumor is really classified into high risk and non-high risk neuroblastoma. So the high risk has a very lower survival probability compared to non-high risk. And really one important problem is that after the tumor of a child is classified as a high risk neuroblastoma, there are no methods to determine whether that disease will progress or not. So measuring, as I said, the mesenchymal cells may provide prognostic ability to the research. And mesenchymal cell quantification can be done by sequencing of epigenetic profiles. So clustering consists on the identification of homogeneous groups among a set of heterogeneous items. In the specific case of neuroblastoma, what I do is I classify cell lines or tumors based on their gene expression profiles. The other thing that I have worked with is classification of SARS-CoV-2 based on their mutational profiles. You would agree with me that mutations are important in cancer and are also important in COVID-19. So for neuroblastoma, this is an example of what's going on right now in the field, because what people try to do is to classify these adrenergic cells and the mesenchymal cells. This is a machine learning method called hierarchical clustering. So we can basically separate, classify, or clusterize the adrenergic and mesenchymal cells. We can do the same using what are called wet lab methods, such as a Western blot. This is a biochemical method of protein quantification. And you can use some scoring systems to classify adrenergic and mesenchymal cells based also on their gene expression profiles. This image here in the right is a similar method. This is also a hierarchical clustering method, but this is based on enhancer peaks or enhancer cis-regulatory regions, the enhancer regions that we use to classify or differentiate the adrenergic and the mesenchymal cells. So what I do currently is I basically score tumors. So what you see here in the left are 498 tumors classified with a high mesenchymal score, top high, second highest mesenchymal score, third highest mesenchymal score, and fourth highest mesenchymal score, which is basically the lowest mesenchymal scores. And what we can demonstrate or see is that tumors with high mesenchymal scores, they have a lower survival probability. Here I'm looking at all risks, but there is some evidence that when I look at the high risk cohort, that cohort also has a lower survival probability. And so this is showing some evidence that the classification of tumors based on their epigenetic profiles may allow some prognosis analysis in high-risk neuroblastoma. And this is very important because, as I said, for high-risk neuroblastoma, there's no such a prognosis system yet. And that's what we're working with. So I am going to end now my talk by showing you one thing that is really interesting in my opinion, which is the fact that we can compare these very different types of diseases. On the right, I have a heat map of neuroblastoma. This is using the RNA-seq profile of neuroblastoma. Sorry if I said this is the left, this is the right image. In the left side here, we have a heat map showing the mutational profile of COVID-19. So with the high school students that I told you, we developed this classification system of COVID-19 based on the mutational profiles. So here what's interesting to note is that by looking at the frequency of these mutations, we see some sort of clusterization of the mutations that are in the extreme east of the Earth, such as the samples collected from China, also present in the bat and the pangolin, which are thought to be the animals that are more permanent hosts of the virus. So really here, what I want to say is that computational biology methods allow us to compare or to have a sense of common analysis that can be done in two completely different diseases, such as neuroblastoma and COVID-19. So in conclusion, as I said, bioinformatics can be used to study and understand these two very different diseases and many other types of different diseases as we saw during this presentation. And more specifically, in my case, bioinformatics may allow the development of prognosis system for high-risk neuroblastoma. So I would like to end now. Thank you so much for listening.